Good morning, everyone. Today, we're joined by Michael Cole, professor in our Department of Microbiology and Immunology, a member of our Georgetown community for 36 years. Michael, thank you so much for joining us. We're so glad to have you for this conversation. Let's jump right in. As a professor of microbiology and immunology, you've taught courses on topics ranging from infectious diseases, the study of the immune system, bacteria's pathways of infection and cause of disease. Could you share with us a little bit more about these topics? What's driven your excitement and your focus as a scholar over all these years?
I think that what started it all for me was, as you know, I started life as a dentist, and the two principal diseases in dentistry are dental caries and periodontal disease, and they're caused by resident members of the oral microbiota. So I was attracted by trying to understand the interaction of those bacteria with the immune system and with the ecosystem in the mouth. And at that time, or shortly before I started to become interested in that, the whole area of mucosal immunology broke open. The principal antibody that functions at the mucosal surface, secretory IgA, had been discovered only a few years before, and it was slowly becoming recognized that the mucosal surfaces had a completely unique immune system. And so the combination then of this unique immune system and these resident bacteria that were causing disease led me into trying to understand how those two were related. What was the, how did the immune system regulate those microorganisms, if at all? And I was very fortunate to be able to postdoc at the University of Alabama, Birmingham, where they had arguably one of the best labs on mucosal immunology at the time. And that was the sort of beginning of it all, I suppose. And that's what I've been sort of doing ever since, really, you know. And that led me in, really was a natural extension to then looking at pathogenesis and the interactions of the immune system with infectious agents. I think it all came out of that, really.
Got it.
Yeah.
Well, as you're talking about immunology, as we think about the developments of vaccines for SARS-CoV-2, the virus responsible for COVID-19, what can our understanding of the immune system tell us about immunity and the body's process of achieving immunity in response to vaccination?
Yeah, that's an excellent question. And I think that one of the things one learns when you look at all of these myriad agents that infect us, bacteria, viruses, fungi, parasites, that they're all attacking the same immune system. And guess what? They're all doing it in pretty much the same way. So I think with SARS, we've been able to build on a lot of our knowledge of how the immune system controls viruses. I think there have been some seminal findings that have contributed to this. One is the discovery of how important the dendritic cell is to instructing the adaptive immune system as to the presence of and the type of infectious agent that this cell can tell a lymphocyte This is a bacterial pathogen. This is a viral pathogen. This is a DNA virus, an RNA virus. It can tell the immune system, is this pathogen outside the cell or inside the cell? And I think our understanding of that is fundamental to the whole of vaccinology, if you like. And then I think an increasing understanding really of that axis of the dendritic cell and the T lymphocytes and B lymphocytes that make cytotoxic killer T cells and B cells that make antibody have all sort of meshed together, if you will, to give us a really, I think, good understanding now of how the immune system sees these pathogens and how it decides on which are the most appropriate mechanisms to defend against them. And the other side of that is, of course, the pathological effects of the immune system. So we've learned that inflammation, while essential to host defense, and I suppose the Romans and the Greek physicians talked about inflammation, Galen and so on, but to understand the basic biology of inflammation and how inflammation is a two-edged sword, as we see with the adverse effects of inflammation in COVID and in other diseases. And I think what I always try to impress upon the students when I'm sort of talking about this material is to try and point out to them just what we discussed a little bit earlier, is that many viruses do these sorts of things. Even some bacteria do these sorts of things. So it's not as if SARS is really unique in many respects, because we've seen these sort of actions or results of infection with other infectious agents over the years. But I think we are beginning to sort of unpeel it more and more. Mm-hmm. And yeah, it's fascinating, really.
Yeah. Now, you also serve on the faculty of our Masters of Science graduate program,
right,
in biohazardous threat agents and emerging infectious diseases. Can you share with us a little bit about the program and the kinds of career paths and opportunities our students consider following their time in the program?
Yeah, I'd love to do that. I have to give credit to Dr. Erin Sorrell, who's the current director of the program, and she's really, I think, taken this to another level. So hats off to Erin. She's done a fantastic job. Yeah, this is a wonderful program, and I think it's strengthened. One of its strengths is just our location. We're here in DC. We're close to seat of government, the FDA, the NIH. Fort Detrick is not too far away. And of course, we have all of the policy being made here. And so we've been able to incorporate all these various aspects in this program. So we can call on individuals to come from FDA, from Department of Defense, Homeland Security, and so on. And I think that's what makes it a very unique program. The students that have graduated from the program have done exceptionally well, and they've gone into all sorts of areas. About 40% of them have gone into the government and policy, but they've also been in business, biopharma, academics, consulting, and healthcare. So I think that there's no limit really to where you can go after taking this program. And it really, I think, has been a great success for the university and so on. So hopefully that it will continue to thrive.
No, thank you. Thank you. Now, you authored a textbook just 2 years ago, 2019, entitled Unifying Microbial Mechanisms, Shared Strategies of Pathogenesis. And you're also the co-author of the text Case Studies in Infectious Disease, which is an overview of some of the most common agents of infectious disease worldwide. Can you talk about the process of developing textbooks such as these? What are some of the approaches you use to help students grasp these areas of study?
Well, starting with the case studies book, I think when the co-authors and I sort of sat down to think about that book, we were wanting to have a concise book because it was targeted largely towards medical students, not exclusively, but that's a large audience for the book. And also, we wanted it to be of worldwide application. So we made, with our choice of agents, we made sure that we covered many of the diseases from the developing world, as well as agents that are common in the United States and in Western Europe, for example. And we went for a sort of boilerplate design so that every case in the book conforms to the same style and the same progression, which I think makes it easy for the students. We included the essential sort of biology of the organisms, as well as the clinical infections by them. And we had a number of, at the end of each chapter, we had self-study questions that they could answer. And also the book was designed to go online. So we had that concept from the very outset of the book. And it's proved to be quite successful. We're just in the process of the second edition of that book. So I think that was really the thrust, if you will, with that book. With the pathogenics text, that was came to me, not in a dream, but came to me as a result of teaching pathogenics in the department, and in many departments, it's taught the same way. I saw that teaching the subject sequentially, well, that's what we would do. We'd start off with bacterial pathogenesis and viral pathogenesis and fungal pathogenesis, and yet the same immune system was being attacked by all of them. And it seemed to me that, and it was clear that the same sort of methods of evading the immune system were being used by all of them. So I thought to myself, it doesn't make any sense to do this, because what would happen is the students would go in, they'd hear about bacteria and the way that they evaded the immune system. Then they go to viruses, see how they evaded the immune system. And it was the same, so complement and, B cells, T cells. So I thought, no, let's do this in a completely different way. And so that's what I set about to do. And it was sort of unique. I don't think there's another book, or had been another book really, that sort of approached it like that. And the other thing I decided was to take all the immunology out of it, because that's, most pathogenics textbooks are half immunology. So they teach an immunology course before they get into the pathogenics. And you shouldn't really be taking pathogenics until you're grounded in immunology and microbiology. So I cut that out of it and said, there's so many good textbooks in immunology, I don't need to, repeat that. And so I focused completely on the pathogenics. And And there it was. And that's the way I teach the course now in the department. So we'll see, I suppose, how, how that works out. So far, so good.
Thank you. Thank you.
Yeah.
Now, you mentioned a few dimensions of your journey. You mentioned your postdoc at UAB and
Right.
your start as a dentist. I'd like, if you could, you attended university in London. You completed
Right.
training as a dental surgeon, now 36 years at Georgetown.
Right.
Looking back from today, tell us a little bit about your path to Georgetown and how have you seen these fields change since you were a student?
Yeah. Yes, I sort of, as you said, sort of took a bit of a career deviation, I suppose. I think what I've seen happen, and I'm not entirely comfortable with it, but I've seen the move away from the so-called generalist. And I don't mean that in a disparaging way. But when I started out, the scientists of the day were much more broadly based and had a much broader knowledge base of their area and other related areas. Now, of course, people might say, well, there wasn't as much knowledge then as there is now. But I think what we've seen is a inexorable progression to the specialist and the ultra specialist. And I think that while that has its strengths, clearly, it also has its weaknesses in the sense that often they only see a very small part of the picture. And I think one can see that fairly clearly in microbial ecology, for example, or the microbiome, which, if you know, is huge now. Everything is to do with the microbiome. I mean, the advances in molecular biology have made it very easy now to do huge surveys of microorganisms in the human body. But so often, what's missing from all of that, the cataloging, is the actual environment, the ecosystem to which these bacteria belong. Because basically the microbiome is focused on the bacterial microbiome. And so you find that, yeah, there's a lot of cataloging, there's a lot of, we found this and that, but actually when it comes to saying, well, what does this mean in the broad picture of the ecology of the large bowel, or, you know, that's missing, because unless you have, and I think the answer to someone will say, listening to this, well, that's why you have team research. You know, you have your microbiome, you have your ecologist. And I think that, yeah, that's true. That's the way that you can overcome that. But frequently that isn't done. Whereas in the previously, I think we might not have known as much. We had I think we had a better idea of what we did know, how it all sort of fit together, if you will.
Sure. Thank you. Thank you.
Yeah.
And thanks for taking the time for this conversation. It's great to be able to spend this time together.
Yeah.
As we bring this conversation to a close, is there a message you'd like to share with our university community?
Well, as you said, I've been here for 36 years. And I was thinking about really what were the sort of most important rewarding aspects of being a member of this community. And I think it's being a member of this community. I think one of the greatest things that I did was becoming involved in the governance of the university, joining the faculty senate, first at the caucus in the medical school, and then going on to the main faculty senate. I came to appreciate the wealth of knowledge, the excellence of the faculty. I got to meet people that I would never have met if I'd have stayed up in the medical school and not done that. Met some wonderful people, served on a lot of committees where you met everyone from everywhere in the law school and business school. And to me, that was a real revelation and a really rewarding aspect of it. And I thought, what if I'd never done that? You know, what if I'd have just stayed up in my lab and not done that? And I think I would have missed a tremendous opportunity. I mean, after all, that's where I first met you.
That's right.
So that can't be bad.
You're very kind.
Yeah. So, you know, I look back on that with a great deal of fondness. And yeah, and the other thing that that always comes back to me is Georgetown is a place where you learn how to think. That sounds sort of pretty mundane, but actually pretty important. Yeah, I'm very happy that I came here, and that I've spent a lot of years here, about half of my life, actually. So, yeah.
Right. You know, Michael, I know you're preparing for retirement soon, and I just want you to know how much we appreciate all of your contributions over the course of all of these years. I too remember very well when we worked very closely together in the 1990s in our process of shared governance.
Yes.
It was a joy then, and it's a real privilege to be able to spend this time together with you now. And you have my very best wishes as you take this next step in your own journey.
Well, thank you so much for inviting me, and I've really enjoyed it.
Thank you, Michael. And thank you all for joining us for this conversation. I look forward to being with all of you again soon. Take care of yourselves and take care of everyone around you. For every Hoya, everywhere.